research is focused on understanding how plants sense freezing and how they adapt their membranes to survive it. I think a lot about a product. I think a lot about how does what I'm doing today in Arabidopsis, how will that eventually, maybe impact a crop, or how will that eventually give us a product that a farmer can use. There's been the predictions that we have to feed 9 billion people by 2050. And so I think if we understand freezing research, we can potentially plant our crops a little bit earlier or have them better prepared for, like, if we plant early and have a late season frost, that would be really helpful. So I do a lot of lipid biochemistry, and specifically most of my project works on a model plant called Arabidopsis. And we like to use it because it grows really quickly and it has a lot of genetic resources. But recently some of my projects have started reaching out and looking at crop plants or other plants that are known to have the protein that I research and looking at how it's regulated in those species also. Most of what my research is on is understanding how membranes change. So membranes, specifically what we focus on the chloroplast a lot, right? So the chloroplast is really important for photosynthesis and for powering the plant. Membranes are sort of what keep cells together and keep certain compartments separated. And membranes are made up of proteins and lipids. Lipids are made up of a head group and 2 fatty acid tails. We focus on how those lipids change in response to freezing, and we want to try to understand it so that we can maybe take advantage of some of these mechanisms that plants already use to help work on engineering plants that are less cold or less freezing tolerant, or make it so that plants can respond more quickly so that we can sort of fight off some of the damage that happens during freezing. There's actually a lot of similarities between what happens to a plant when it's frozen and what happens during drought, because there's big water potential changes in the plant. I've always had, science has always been a big part of my life, and I guess if you're involved in 4-H, you know that a lot of the write-ups that you have to do are really training you to think like a scientist, right? You have to have a hypothesis, and then you do your project, and then you write about what you learned. And that's a lot of what I do every day. I think about how will my project impact this, or what part of change, what changes do I need to make to my project, and then what do I learn from it, so I reflect on that later. I think the most exciting part about being in science is that, you never know what the results are going to be. So you may have this experiment and you think it's really well planned out, and then the plant throws you a curveball and you totally have to adapt and reorganize your experiments and reorganize how you think. And so it's a challenge every day, and that's what I think I like the most about it.